Okay, so what we're going to look at now is we're going to look at genome projects and making DNA fragments. So we looked a bit at DNA fragments the other day. I'll point out when to go back to that recombinant DNA video to have a little watch of those if you can't really remember all the ins and outs of certain parts of that. So gene technologies, we need to understand the whole genome to be able to do gene technologies. And gene technologies, as we were talking about the other day, we were able to actually go in and we can splice out parts of DNA, we can take bits out, put DNA from one organism into another, and lots of other different techniques. But to be able to have done that, we needed to have basically had to be able to sequence a whole genome first, and that's what we're going to have a quick look at. Start off with. So genome sequencing, you'll have covered some genome sequencing in the Human Genome Project in GCSE. And what we need to be really, really clear about now at A level is that our full genome sequencing involves not only our nuclear DNA, that's the thing that we normally go to originally. We go, okay, yeah, DNA sequencing, where's our DNA? It's in our nucleus, our nuclear DNA. But we also, as you know, have DNA inside our mitochondria, and in plants, in chloroplasts and mitochondria. So when we do a full genome sequencing, we have to also include DNA that's in mitochondria and chloroplasts if the organism has chloroplasts. So that is a really important point to take away from this now at A level, is that our full genome sequencing involves nuclear and mitochondrial and all chloroplastic DNA. So all our genes in an organism. And as technology has improved over time, we've not only just sequenced human genomes, we've been able to sequence bacteria, lots of different plants, lots of different animals, and being able to do that has been great because it's allowed us to make comparisons between individuals of the same species. And we might look at two humans who've got two different phenotypes, and then we could go back to their DNA, look at their genome and actually compare them. Well, why are their phenotypes different? Which gene is it that's causing it? Which genes are different in these two individuals? But we can also then make comparisons between different species as well. We've used it a lot for evolutionary relationships. So evolutionary relationships that we've had in the past were very much based on phenotypes of bones that have been found and skeletons, and we've looked at them and gone, well, this one sort of looks similar to that one. And then we had carbon dating, so then we were able to go, all right, well, this one's this old and this one's this old, so this one came first, and then it's obviously evolved and it's got a different knee joint and it's evolved this different knee joint, and so this animal's come from this one. Having the Genome Project, we're actually able then to look at evolutionary relationships in a different way. It's actually dramatically changed a lot because some individuals might have very similar phenotypes and because of niches. So they've evolved to suit a specific niche, and actually they might have very similar phenotypes, but their evolutionary history is actually very divergent and they don't come from a close evolutionary ancestor. So that's actually quite changed lots of evolutionary relationships that we've thought of in the past. And then some individuals, some different species might be more closely related than we thought because they're so phenotypically different, but genomically they're very similar. And that's just all to do with obviously how evolution works for an individual to prosper in their niche. It also allows us to have differing responses to medical treatment. If we can look at someone's whole genome, we can maybe actually see how they're going to respond to different medicines, different drugs, how their body is affected by different illnesses. So if we can look at a human's genome, we can actually treat them differently and have this more specialized, individualized medicine. Now, in an ideal world, that'd be fantastic and we'd be able to do that for everybody. But at the minute, that isn't something that's doable in medicine. It's not that we can't do it, it's just too time consuming, too expensive, and we just don't have the manpower really to do that. But it's not that it's not actually physically possible. So when we do gene sequencing and we're sequencing a whole genome, we do this, we can only work with fragments of DNA. So gene sequencing methods only work on fragments of DNA. So if we want to sequence the whole entire genome of an organism, so let's say, for example, a chimpanzee, we actually have to go and chop up all that DNA first. and the smaller pieces are sequenced, and then we have to put them all back in order to give the sequence of the whole genome. So when the first, so the Human Genome Project, which you'll have heard about when you were doing GCSE, that was completed in 2003, and that mapped the entire sequence of the human genome for the very first time. That took years and years and years because technologies for the first time of anyone doing it, and they didn't have anything else to go off. So they had to basically break up all the DNA into smaller pieces, sequence them, put them back in order. So that took a long, long time. Now, sequencing of genome is actually really, really quick, and technologies have got super fast at doing it, but it's really important to remember. But yeah, it took a lot longer to do that. Something that I'm just going to have a quick chat about is simple organisms compared to complex organisms. So sequencing the genome of simple organisms helps us identify their proteins, and that's really, well, for starters, that is really useful. So the proteome, this is a word that you might have heard of before, you might not have. Proteome. So our proteome of an organism is all the proteins that are made by it. So this is all the proteins made by the organism. That is a proteome. So genome, all the genes present in the organism. Proteome is all the proteins made by an organism. If you think back to first year of A-level, some parts of your genome code for specific proteins. So we have those introns and extrons. Some don't code for any specific genes, some do. Then the genome codes for specific proteins. So we have that coding and non-coding part of the genome. So the genome that codes for these specific proteins is what we're sort of looking at. It's really important actually in these simple organisms when we've sequenced them. So simple organisms such as bacteria, for example, they actually don't have much non-coding DNA. They have very, very little non-coding DNA. So it's really, really, really easy to determine their proteome. So they have little non-coding DNA, whereas we have lots of non-coding DNA. They have very little non-coding DNA. So it's really super easy to be able to sequence their proteome. And their proteome from the DNA sequence of their genome. So the genome is just literally all your bases, or all your DNA nucleotide bases that you have in your DNA. The proteome is then, we get a genome and then we can work out the proteome from the genome by actually looking into it and going, okay, this section codes for a gene, this section codes for a protein, this section does, these bits don't. So bacteria are great because we can actually work out their proteomes really, really quick because they have very little non-coding sections. Why is this useful? Well, it's really, really useful for things like medical research and development. So if you're thinking about bacteria, they make us ill a lot. So this is really good for finding, for example, something like a protein antigen. So I'll just pop this here and I'm writing all over here. Protein antigen. So as you can remember, protein antigens are found on the surface of disease-causing bacteria. We also get them on disease-causing viruses as well. And so if we can code for these and find the DNA that codes for them, it can help us develop vaccines to prevent the disease. So an example would be the N meningitis group B bacteria, and that causes meningitis B. So the sequencing of the genome of these bacteria actually helped researchers identify antigens for use in developing a vaccine against the disease. Really, really, really important. Just a quick recap about vaccines, just a reminder. Vaccines contain antigens that cause your body to produce memory cells. If you're later infected by the pathogen with the same antigens, your memory cells will recognize it more quickly and divide to produce antibodies against it more quickly so you don't get sick. You are still infected with the disease, but you are able to fight it off a lot quicker so you don't become ill with it. And that's, again, if you think about COVID at the moment, lots of people get very confused about that and go, oh, I can't get ill. I can't get COVID. I can't get COVID because I've had the jab. You can get COVID. You might not get sick from COVID because you've had the jab. So your memory cells work so that then you can fight off infection quicker and faster and make those antibodies quicker and faster. But you can still get COVID. COVID can still infect your body. So as you can probably guess, I've said bacteria, really easy to get their genome because they've got very little non-coding DNA. More complex organisms, for example, a human, they've got huge sections of non-coding DNA. So they are a lot more complicated. They've also got regulatory genes. So these are the ones that code for particular proteins that should be switched on and off. So when we've talked about epigenetics, they've got all that coded in there. So it makes it really hard to translate their genome into the proteome. And if you think, these are going to be harder because they're not these single-celled organisms. They're multicellular organisms. So we're going to have all these regulatory genes switching things on and off. So it's hard to get their genome into their proteome because we've got to sift through lots and lots of non-coding DNA, sift through lots of regulatory genes. So because there's so much non-coding DNA, it's really hard to find the bits that code for proteins among all this non-coding DNA and regulatory DNA. However, work's been done on the human proteome, and the codes for more than 30,000 human proteins have been identified so far. So whilst we've got the Human Genome Project, we don't have the full Human Proteome Project. So sequencing methods, we've done a little bit about sequencing methods, but I'll just do a quick recap now. It was super labor intensive to begin with. It was really expensive and it could really only be done on a small scale. So like the Human Genome Project, they just did that one genome of one human. Now the processes are automated, they're a lot more cost effective, and we can do them on a large scale. So there's something called pyrosequencing, and that's really new, recently developed technique. And so that can sequence about 400 million bases in a 10-hour period. So that's really fast compared to old techniques. So as we get these newer and faster techniques, such as pyrosequencing, and scientists are able to sequence a whole genome a lot quicker. So we're just going to have a quick look at something called a mini satellite. So we've got on here a chromosome. And as you know, all diploid organisms, such as humans, have 2 copies of their genome in every single cell. And this is divided into chromosomes, and chromosomes come in pairs, 23 pairs in humans, one half of the pair coming from the egg cell and one half of the pair coming from the sperm cell originally. And so each of which of these chromosomes contain up to hundreds of millions of nucleotides in a single coiled strand. So then if we look into the DNA even closer, chromosome even closer, we get our DNA strands. So along the DNA strands are our sequences of nucleotides, or our bases that codes for our proteins. So these are the organism's genes. Obviously in there, we could have our non-coding sections. Introns. In addition to genes, organisms can contain many repetitive non-coding base sequences. So non-coding regions of DNA are called introns. The coding are called exons. They are non-coding because, unlike genes, they are not translated into proteins during protein synthesis. I've mentioned this in the past. We used to call it junk DNA. We don't call that anymore because actually we think it has it does a lot of good. It does a lot of work, and we just don't really fully understand it anymore. Junk, we basically would infer that we don't need it and we can get rid of it, but that's not true. It has a lot. This non-coding areas have a strong play in the epigenetics, and there's a lot we can look into with that. Then we get something called a mini satellite. So inside these introns, inside these non-coding areas, we can get these short repeating sequences of bases, and these are called core nucleotide sequences, and these are called mini satellites. Sometimes called hypervariable regions, but you'll mainly hear them called mini satellites. And they're hypervariable because they vary considerably between individuals. So the uniqueness in our DNA is actually in these non-coding regions, which is really interesting. And so these mini satellites are what we can actually use to sort of compare one individual to another a little bit more, because they're really, really variable. And there's so difference between individuals, even of the same family, and you can then start to pick out individuals quite quickly like that. And these mini satellites, they vary both in the number of repeats that they have of these repeating sequences of a core nucleotide and the base sequence of the core nucleotide sequences. So the probability of two individuals having the same many satellites is really, really low. And this is a feature of genomes that is exploited in genetic fingerprinting. So when someone says, oh, we've got a DNA match at a crime scene, it's these mini satellites, it's these repeating sequences. It's that we use for genetic fingerprinting. So that's what we're going to have a little quick look at now. So a genetic fingerprint is created by looking at an individual's mini satellite. The choice of mini satellite is really, really important. So the profile needs to choose those mini satellites that show the most variation, because there is a very, very small chance that two individuals could have matching fingerprints. So we need to pick the ones that have got the most variation in them. So we call it genetic fingerprinting because we basically use the idea of normal fingerprints. So for hundreds of years, we've been able to identify people from their fingerprints. We all know that every human individual's fingerprints are unique to them, but we can now use their DNA instead. And again, like we said, not all of an organism's genome codes for proteins, and we use these mini satellites, hypervariable regions, sometimes in AQA they call them as well variable number tandem repeats. I'm just going to pop that in here actually, and you'll see it abbreviated as VNTRs, variable number tandem repeats, base sequences. And like I said, they do not code for proteins, and they repeat next to each other over and over and over, sometimes thousands of times. So you could get something like this, and it just goes again and again and again. So you could get CATG, 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 and it just goes again and again and again constantly. So that's the repeat of the non-coding base sequence of CATG. The number of times these sequences are repeated differ from person to person. So the length of these sequences and nucleotides differs too. So a 4 nucleotide sequence might be repeated 12 times in one person. So they'd have 48 nucleotides, but this same 4 sequence nucleotide sequence could be repeated 16 times in another person. So they would have 64 nucleotides. So the repeated sequences occur in lots of places in the genome. And the number of times the sequence is repeated at different places in the genomes can be compared between individuals. And this is what we call that genetic fingerprinting. And again, I'll reiterate, the probability of 2 individuals having the same genetic fingerprint is very, very low, because the chance of 2 individuals having the same number of variable number tandem repeats, mini satellites, at each place they found in DNA is very, very low. So let's just move all this off. So the main stages involved in genetic fingerprinting are these. Extraction, we need to extract the DNA. Digestion, separation, and hybridization. And then finally development. So we're going to look at those stages, and we'll have a bit more of an in-focused look at these in a bit. So that's the basis of how they have these 5 main stages. So we have to extract, digest, separate, hybridize, and then develop them. So this is now looking at how we take this genetic fingerprint. So it's a banding pattern that's produced from an individual's DNA that is unique to that individual. So you'll see them, and I'll get you some images in a bit where it shows them, and they look like bandings on a picture. And it is all just to do with those variable number tandem repeats and how many times they're repeated. And it's produced from individual's DNA that's unique to that individual, and it can use to identify samples such as those found at a crime scene or in the establishment of paternity. So when we do paternity tests, we don't like compare full genomes. We literally look at these variable number tandem repeats, mini satellites, and say, yeah, that person's the father, that person's the mother, and that's how it works. So what do we do first of all? So we go through collection. A sample of tissue is taken, which contains the DNA that needs to be analysed. It can be lots of different things. It could be blood, cheek cells, semen, or hair, or any other suitable sample of individual cells. So for example, if it's a crime scene, if there's some blood, we would take it from there. So there's lots of different things that we do. So in that image, a buccal swab is being taken cheek cells, and it has to be quite rough on cheek cells to actually get the cells off rather than just saliva, but we can get it from saliva as well. And then we extract it. Basically, the process that we use in genetic fingerprinting is something called PCR, and we will look at PCR in a bit more detail again. Later, we'll have a whole session on PCR and how PCR works. PCR is polymerase chain reaction, and in that we'll go through every single little stage, what we add at each stage, what temperatures we do things at. But at the minute, we'll just do a brief overview. And we use PCR to make many, many copies of the areas of DNA that contain those variable number tandem repeats. So we basically want to just get rid of all the rest of the DNA, and we just want the bit of DNA that we want. So we use primers to bind at either sides of these repeats, and so the whole repeat is amplified. So what we do is we extract. So we get the sample and we mix it with phenol and chloroform, and we centrifuge it. And centrifugation, if you can remember, where we pop it into a machine, which is a centrifuge, and it gets spun at a really high speed and it mixes it. And that mixing actually causes separation, and the proteins precipitate out. So as we can see here, we've got an aqueous water layer, and we've got that precipitated DNA strands being collected with a hook at the top. So these are all the DNA at the top. And so the DNA literally will bubble up. Sometimes it can be sat at the bottom, and you can basically, and you'll have a globular, visible to that naked eye specimen of DNA in there. So when you add the sample to the phenol and chloroform, you cannot see it with your naked eye. But once it's been around centrifuge, you can see it. You can actually see the DNA there, and it the proteins precipitate out, and obviously DNA is a protein. So the DNA is then precipitated out of the aqueous water layer using ethanol, and you can actually physically extract it as well. So then it goes through something called digestion. We only want those areas that have got those variable numbered tandem repeats, as mini satellites. So what we do is we use restriction enzymes to cut the DNA at points close to, but not in, those mini satellite regions, allowing these to keep their original lengths characteristic to the individual being testing. So again, that's really, really important that we use, sometimes we call, so these restriction enzymes are also sometimes use the word name primers. So you can hear restriction enzymes and primers used interchangeably. They cut them at the points, and then we don't want to cut them at the point because then we could lose stuff. So we have them very, very close to those regions that we want to test for. And so what we will do is we'll end up with DNA fragments where the length in nucleotides corresponds to the number of repeats the person has at each specific position. So one person might have 80 nucleotides, another person 120. So if we go back to that CATG, if we cut for that CATG repeat, length is a repeating course sequences after digestion. So you might have different lengths. We might look for the same person at a few different repeating VNTRs and look at their differing lengths. So once we've digested them, we're going to separate them. But actually, I'm just going to go back here before we even separate them. Before we separate them, we add a fluorescent tag to them. And this means that we can then view everything under a UV light. So that's really important. We add a fluorescent tag at this point so we can view them under UV light. So then the DNA fragments go under something called electrophoresis, gel electrophoresis. And basically, the overview is the digested fragments are going to be separated by size, by electrophoresis. The gel is then immersed into an alkali solution to separate the DNA into single strands. And this is denaturation. So what happens is the DNA at the top, as you can see where these pipettes are, is the DNA mixture is going to then be placed into these wells. And this is a slab of agar gel. And we then cover this gel, which you make yourself in the lab, in a buffer solution. And that buffer solution is able to conduct electricity. An electrical current then is passed through. Yep, it's still here. Is going to be passed through the gel, and the DNA fragments are negatively charged. So they move towards a positive electrode. So basically the negative electrode is at the bottom where the DNA is, and they're going to move up to this positive end up here. So we move towards a positive electrode at the far end of the gel. Our small DNA fragments move faster and they travel further through the gel. So the DNA fragments separate according to size. So the smallest is going to be closest to the top, biggest closest to the bottom. We then need to actually view this. So the DNA then gets transferred to a nylon membrane, and we use blotting paper to draw it up out of the gel by something called capillary action. This process is called Southern blotting after Edward Southern, who invented it, and it is really, really hard. And it's why we use PCR to get the samples of blood and stuff and increase the amount of DNA we have, because you can guarantee, especially when you first start working in labs and doing this sort of work, it's not going to work the first time you do it. I've done lots of Southern blotting in my time at university, and yeah, you can get a mess, basically. You can have done everything right up until that point, and you try and do Southern blotting, and it can end up an absolute mess, and then you've got no results. So, yeah. And so then Southern blotting, you can then look at it and view what you can see. So the membrane is immersed in a solution of DNA probes, and we're going to have a whole session on DNA probes later, which hybridize with the chosen core sequences. And the unbound probes are washed off, and the membrane is then dried. And then an X-ray film is placed over this membrane, and the film is fogged in the areas where radiation is present, producing an autoradiograph and the distinctive set of bands known as a DNA fingerprint. How I'm saying we add fluorescence, you can either do this with the X-ray film, or you can view the bands under UV light, and that gives you the genetic fingerprint. So they're the two ways that we can view them. We often use the UV light one more now, just because of the harms of X-ray, and also it's just easier and creates less issues. And then we can get two genetic fingerprints and we can compare them. If both the fingerprints have a band at the same location on the gel, it means they have the same number of nucleotides, and so the same number of variable numbered tandem repeats, mini satellites at that place, and it's a match. So we can use genetic fingerprinting to determine genetic relationships and determine genetic variability within a population. So we'll look at that in a bit. So a profiler can then inspect that genetic fingerprint that we've made, and we can look at it by eye to make a really quick comparison. So the process can also be automated, and a computer using the marker bands can calculate the size and distance traveled by the bands and profile them as well. It is sometimes necessary to consider the odds that somebody else in the population has the same DNA fingerprint as the one risk of a false crime, for instance, to assess the risk of a false criminal conviction. I'm going to quickly go through these last few bits and then go in a little bit more deeper about genetic fingerprinting. So a quick look at the Human Genome Project again. Human Genome Project, it was internationally public funded to sequence the 3 billion bases in the 20,000 to 25,000 genes of human genome. It took from 1990 to 2003 for the first full sequence to be published. And then it was then later corrected and analysed, and then analysis has then been added to results over the following years, and people still add analysis to it today. One of the subsidiary aims of the Human Genome Project was to study the ethical and social implications of a project and to predict and discuss the legal issues that would arise. These questions can come up a lot in the exam, and you can talk about ownership of DNA and eugenics and designer babies. And here we go with something that would be quite good that came out of the Human Genome Project, and that was designer drugs. Drugs can actually be designed now using the knowledge of protein structure, and we gain that from the gene sequence information. So this actually reduces the need for trial and error when we're making these new drugs, and allows doctors to tailor specific treatments to their patients depending on the exact nature of their condition. Designer drugs can be used to treat the fundamental causes of a disease rather than just the symptoms. So this is what we've already done, so I don't want to do that again. So just want to pause this here for a second. So last little bit now, I just want to look in a bit more detail at genetic fingerprinting and what we can use it for. So like I said earlier, the first thing we can use it for is actually for, let's zoom in a bit, determining genetic relationships. So what's really, really important to know is that we inherit these VNTRs, these mini satellites. Oops, we've just gone a bit rogue there. So we inherit VNTRs. So I'm just going to put VNTRs to just make it a little bit quicker, so we can get through that a bit faster. So we inherit VNTRs. We inherit these VNTR bases from our parents, and roughly, as you can probably guess, half of the sequences come from each parent. This means the more bands on a genetic fingerprint that match, the more closely related, genetically similar two people are. So more bands matching equals closer genetic relationship, basically. So example, like I said earlier, would be that you could use for this sort of thing would be paternity tests. The paternity tests are used to determine the biological father of a child by comparing genetic fingerprints. If lots of bands on the fingerprint match, then that person is probably the child's father. The higher number of places in the genome compared, the more accurate the test results. So if you don't compare as many places in the genome, it's less accurate. So that is also something to take into account. So we can use genetic fingerprint to determine genetic relationships. We can also use it to determine genetic variability within a population. So genetic variability in population, this time we're looking for bands to not match. So the more bands that don't match, the more bands that are not matching on a genetic fingerprint means the more genetically different people are. This means you compare the number of repeats at several places to find how genetically varied the population is. So more bands not matching equals greater variation. That is a really simplified version, but we would look at it across the whole genome, and the more places that there's less matches increases the amount of variation. So the more number of repeats that there is at several places, the greater the genetic variability. So like I said, we can use genetic fingerprinting in forensic science. So that is a great place that can be used. So samples from crime scenes, so blood, semen, skin cells, saliva, hair, from possible suspects could link them to the crime scenes. So like we said, again, the DNA is isolated from all the collected samples, so from the crime scenes and from the suspects. We replicate the samples using PCR. The PCR products are run on that electrophoresis gel, and the genetic fingerprints are compared to see if any match. If the sample matches, it links a person to the crime. E.g., a gel that shows a fingerprint from suspect C matches a crime scene, it would link them. We can also use genetic fingerprinting for medical diagnostics. We can use it. A genetic fingerprint can refer to a unique pattern of several alleles. So unique pattern of several alleles, and this is really important. So we can use it to diagnose genetic disorders and cancers, and it's really useful when a specific mutation in the genes isn't known, or where several mutations could have caused this disorder, because it identifies an actual broader altered genetic pattern. So one example that I'm just going to give you here would be pre-implantation genetic haplotyping, so PGH. Pre-implantation genetic haplotyping. And so this is screening of embryos. So pre-implantation, before the embryos are implanted, genetic haplotyping. So this is where embryos that have been created by IVF are screened for genetic disorders before they're implanted into the uterus. So the faulty regions of the parent's DNA are used to produce genetic fingerprints, and these are compared to the genetic fingerprint of the embryo. This wouldn't be something that would just be done for most people going through IVF, because we have to look at what the parents have got, the faults in their DNA to compare. This would be used in parents that have an increased chance of having children with passing on genetic inherited disorders. So something like cystic fibrosis, Huntington's disease. So they might be carriers. So they want to see if that embryo they're implanting, that child would grow up to have cystic fibrosis, for example. So it's a chance to reduce that risk. So if the fingerprints match from the parents' areas that would have faulty regions that would show that they would carry cystic fibrosis, and the embryo would have it, the embryo would have inherited the disorder. So it can be used to screen for things like cystic fibrosis. Another thing we can use it for is to diagnose sarcomas, which are a type of tumor. So conventional methods of identifying a tumor, such as biopsies, they only look at physical differences in tumors. Now, the genetic fingerprint of a known sarcoma can be compared to the genetic fingerprint of a patient's tumor. If there's a match, the sarcoma can be specifically diagnosed, and the treatment can then be targeted to that specific type. Finally, we can use it in, and this would possibly seem a little bit surprising, animal and plant breeding. So genetic fingerprinting can be used on animals and plants, and that is to prevent inbreeding. So this is really important. So we're looking for that genetic variation again. Because if inbreeding occurs, the gene pool decreases. Inbreeding can lead to increased risk of genetic disorders, leads to health problems, productivity, and reproductive problems. So genetic fingerprinting can be used to identify how closely related individuals are. And the more closely related two are, the more similar the genetic fingerprints will be, and the least related the individuals will be then bred together. So this was a session looking at human genome and genetic fingerprinting. We will then have a session at some point about PCR.